Okay, in this video, we're going to talk about our gram-positive pathogenic bacteria, and we're going to split it up into two videos, one that focuses on our Staphylococcus species and one that focuses on Streptococcus. Staphylococcus species are a really great one to start with because we've already covered some of the virulence factors that they have. Staphylococcus are facultative anaerobes. You have heard of Staph aureus and Staph epidermidis. We've worked with both of these in the lab. They are non-motile, and remember they're able to grow in higher salt concentrations, so they'll grow on mannitol salt agar. They are both catalase positive, and we can use this as a diagnostic factor to separate them from streptococcal species. Now, the pathogenesis of Staph aureus in particular comes from three different features. The first, that it's able to evade phagocytosis. The second, that it produces a number of those enzymes that help bacteria invade into deeper tissues. And then the last, that it produces several toxins that we'll talk about. Let's first look at how staph can evade phagocytosis. It produces a protein called protein A, and this protein actually binds to antibodies and prevents them from recognizing the bacteria. So antibodies are really important in helping our immune systems clear or get rid of pathogens, and protein A prevents that from working properly. The second is the production of coagulase. And remember, we attempted to look at coagulase in the lab. This coagulase is actually bound to the bacteria themselves rather than being secreted into the environment. So in a tube of serum, like we can see in our picture, Staph epidermidis, which does not produce coagulase, will leave that serum as a liquid. So when we tip the tube to the side, it'll spread up the tube. Staph aureus, with its bound to coagulase, will cause coagulation to occur, and so we have this kind of gross blood clot at the bottom of the tube. They can also produce slime layers, which are like capsules, only they're a bit looser and more slippery. You can see the nice thick slime layer of Staph aureus here around this bacterial cell, and they can also produce biofilms. And so our slime layer makes them hard to phagocytize, and our biofilm, remember, makes them hard to kill because they are encased in a thick polysaccharide polypeptide matrix. Staph aureus can produce a number of enzymes that allow it to invade deeper or contribute to its pathogenicity. One of those is hyaluronidase, and we already spoke about this one in class, so I'm not going to go over it again, but here's that same figure that we went over in class. Staph aureus also produces a number of lipases, so enzymes that break down lipids, and this allows it to grow on skin and in oil glands where it's naturally found, but where it also causes a lot of infections. And one of the biggest issues with Staph aureus is that they can produce beta-lactamases. 90% of Staph aureus strains produce some form of these enzymes, and what beta-lactamases do is they inactivate penicillin and other beta-lactam drugs like cephalosporin by cleaving the beta-lactam ring that's essential for their function. So these provide antibiotic resistance to things like MRSA. Another set of enzymes we went over in class, coagulase and staphylokinase, produced by Staph aureus. Again, coagulase is an exoenzyme that allows the clotting of our clotting factors around bacteria, and staphylokinase breaks down those clots to allow the bacteria to escape and invade deeper. We did go over those in class, so refer back to your notes from class. Last but not least, Staph aureus produces a number of actual toxins that can have deleterious effects to our cells. The first is that Staph aureus produces a number of cytolytic toxins, or toxins that are going to directly kill cells. These are not very creatively named: alpha, beta, gamma, and delta, and they disrupt cytoplasmic membranes, so they cause cellular membranes to fall apart, which results in death of the cell. Staph aureus also produces a toxin called exfoliative toxin, or exfoliatin, and my arrow somehow got messed up here in translation. But what exfoliative toxin does is it cleaves some proteins that hold the skin layers together. So right along here between these two layers, our purple layer and our pink layer of skin, Exfoliatin cleaves those bonds, and I'll show you a picture of what it looks like later, but it allows that top layer of skin to slough off. And last but not least, Staph aureus also produces enterotoxins. So entero, like enteric organisms, toxins that are going to affect the gastrointestinal tract. These are named A through E, right? So we've already used our Greek alphabet for our cytolytic toxins. Now we're just going to use A through E for our enterotoxins. And these are responsible for Staph aureus mediated food poisoning, which is not something that we hear about all that much, but does happen. Now I apologize for the gross pictures here, but these are some infections that Staph aureus can cause. Scalded skin syndrome, which we'll talk about a bit more in a slide or two. They can cause folliculitis, which is an infection of follicles. They can be relatively small, like in this picture, or they can be large and be called a furuncle or a boil in the picture on the right. If many furuncles come together, they are called carbuncles. Styes, infections there on the eyelid, are pretty common with Staph aureus, but one of the most common diseases of the cutaneous diseases that we see in children is impetigo, which causes these honey-colored crusts on the nose, on the mouth, on the cheeks. And it's highly contagious, and it's spread by direct contact from person to person. Staphylococcal scalded skin syndrome, or SSSS for short. This disease is facilitated by the exfoliative toxin, and again, that toxin cleaves the linkages between the stratum granulosum and the stratum spinosum. So these upper layers of skin, as you can see in this diagram up top, peel off because there's no more bonds, no more proteins that are holding the skin layers together. And on an infected human, it looks like our picture down at the bottom. So it looks kind of like a bad sunburn when a sunburn starts peeling. And these sheets of skin will come off, and the underlying tissues are very raw and very red. We usually see this mostly in infants and very, very young children because the particular type of linkage that this enzyme cleaves is only found in children. And as we age, different linkages form there in our skin. It can be mistaken for child abuse. It may look like this child has received some sort of burn and that's why the skin is peeling off. So a test would be necessary to make sure that it's actually staph and not a burn or a sunburn. And the final staphylococcal disease I'm going to tell you about are those that are systemic, or those that get into the bloodstream. Our other staph diseases are not usually fatal, but this one is, and it's called toxic shock syndrome, which you may have heard of before. It's facilitated by a completely different toxin called toxic shock syndrome toxin. And this toxin gets into the bloodstream and it causes an overactivation of the immune response. So a lot of inflammation, a lot of activation of immune factors that make you feel even more sick. One of the hallmarks of this disease is a red rash, like you can see here on this patient's arm. It's kind of a spotty rash, and it can be accompanied by other symptoms such as fever and vomiting, a drop in blood pressure, and peeling of sheets of skin. Usually this is treatable with antibiotics, but it's still 5% fatal, so 5% of cases still die even with treatment. Now, fortunately, it's really easy to diagnose and treat staph infections. The best way to diagnose is to simply do a gram stain from pus, from blood, from other bodily fluids. And seeing those gram-positive cocci is pretty indicative of Staph aureus. We can take those bacteria we isolate from our patients and run a quick coagulase and catalase test and have a diagnosis really quickly. For the most part, staph infections are very treatable. You can, in some cases, use things like penicillin G or cephalosporins. Again, these are beta-lactam antibiotics, and so our staph may have beta-lactamases that inactivate them. In that case, we can use a different drug called methicillin, and this actually is resistant to beta-lactamases. So those beta-lactamases won't break down methicillin because of its structure. For anything, the best method is prevention, and to prevent a staph infection, because they are in general cutaneous infection, You simply practice good hygiene. Okay, that's it for our staphylococcal species. In the next video, we'll talk about streptococci.